Membranes define external boundaries of cells and organelles. This video will focus on the key components of membrane structure, while a future video will focus on membrane transport. In 1920, physiologists Gorder and Grendel discovered that plasma membrane is mostly composed of phospholipid bilayer. Phospholipids are amphipathic molecules consist of a hydrophilic head group and hydrophobic tails. In the phospholipid bilayer, the hydrophobic tails interact with each other, while the hydrophilic head groups interact with the aquatic environment in our body. In 1935, physiologist Davison and biologist Danieli proposed that the phospholipid bilayer is coated with a thin sheet of proteins. However, the membrane proteins are mainly globular, therefore wouldn't fit in the sandwich model. In 1972, biologist Singer and biochemist Nicholson proposed a new model for plasma membrane that became the most widely accepted model nowadays. The fluid Mosaic model states that the membrane is mainly composed of a fluid structure of phospholipid bilayer, with a mosaic of various proteins embedded within it. Cholesterols are also embedded between the phospholipids. Peripheral membrane proteins are bound to the surface of the membrane, while integral membrane proteins penetrate the phospholipid bilayer. I've covered different types of membrane lipids in my previous video on lipids. The most common membrane lipids are glycophospholipids named after their polar head groups, such as phosphatidylethanolamin, abbreviated as PE, phosphatidylcholine, abbreviated as PC, phosphatidylserine, abbreviated as PS, and phosphatidylinositol 4,5-bisphosphate, abbreviated as PIP2, which plays an important role in signal transduction pathways of both acetylcholine and insulin, as mentioned in the previous video. Each type of membrane has a characteristic composition of lipids. Within a cell, the phospholipid cardiolipins are abundant in the inner mitochondrial membrane and plays an important role in signaling apoptosis, or programmed cell death. Neurons are particularly rich in sphingolipids such as sphingomyelin, abbreviated as SM, cerebrocytes, and gangliosides. Within the plasma membrane, phospholipids are asymmetrically distributed between the two monolayers of the bilayer. Phosphatidylcholine and sphingomyelin are typically found on the outer monolayer, as well as various glycolipids and glycoproteins, which mainly function in cell recognition. Glycosylphosphatidylinositol, abbreviated as GPI anchors, are derivatives of phosphatidylinositol, which bears a short oligosaccharide joined to carboxy terminal residue of a protein through phosphoethanolamine. GPI anchor proteins are only located on the exterior surface of plasma membrane and play an important role in fertilization, embryogenesis, and the immune system. Phosphatidylserine, phosphatidylethanolamin, and phosphatidylinositol bisphosphate are found on the inner monolayer. Besides GPI anchors, other lipid anchor proteins are mostly found on the intracellular surface, including palmitoyl-linked proteins which attach through cysteine or serine residue, myristoyl-linked proteins which attach through amino-terminal glycine residue, and farnesyl-linked proteins which attach through carboxy-terminal cysteine residue. Changes in membrane asymmetry may have significant biological consequences. For example, the externalization of phosphatidylserine on the outer surface marks a cell for destruction by programmed cell death. Peripheral membrane proteins attach to the membrane through electrostatic interactions and hydrogen bonding, and can be easily removed by carbonate at high pH, urea, and chelating agents. Some peripheral proteins attach covalently through lipid anchors. Many peripheral proteins are effectors of signal transduction pathways, such as phospholipase C, abbreviated as PLC, which is activated by G protein and cleaves phosphatidylinositol bisphosphate into second messengers, inositol triphosphate and diacylglycerol. Amphitropic protein is a special class of peripheral proteins that associate reversibly with the membrane. Examples include protein kinase C, or PKC, from the same signal transduction pathway, which is activated by diacylglycerol and calcium ion, and can enter cytosol to phosphorylate other proteins. Integral membrane proteins associate with the lipid bilayer through hydrophobic interactions and are only removable by detergents, organic solvents, or denaturants. They are composed of either alpha helixes or beta barrels. Examples of integral proteins include various membrane transporters, signal receptors, and channels. Most of the phospholipid can move freely laterally, creating a fluid environment. However, phospholipid rarely flip-flop transversely without any catalyst. As temperature cools, membranes switch from a fluid state to a solid state. Membrane fluidity is determined by the saturation of hydrophobic chains of phospholipids. Membranes rich in unsaturated hydrocarbon tails form kinks, increasing membrane fluidity. Cells may regulate their lipid composition to maintain membrane fluidity. The sterol cholesterol acts as a buffer for membrane fluidity. At warmer temperatures, cholesterol restrains the movement of phospholipids, decreasing fluidity, while at cooler temperatures, cholesterol maintains fluidity by preventing tight packing. Trans-bilayer movement of phospholipids are catalyzed by three types of translocators. Flipase is a P-type ATPase that moves phospholipids from the outer layer to the inner layer, driven by ATP hydrolysis. Flopase is an ABC transporter that moves phospholipids from the inner layer to the outer layer, also driven by ATP hydrolysis. Scramblase is a passive transporter that moves lipids in either direction towards equilibrium, which requires calcium ion as cofactor. More details about membrane transporter will be covered in a future video. In an ocean of liquid disordered phospholipids, there are some rigid regions known as membrane rafts, which consist of high concentration of glycosphingolipids such as cerebrosides and gangliosides that contain long-chain saturated fatty acids and cholesterol. The membrane rafts are enriched with GPI-anchored proteins and fatty acyl-linked proteins, either through palmitoyl or metastoyl groups, but not prenylated proteins like farnesyl-linked proteins. One of the palmitoyl-linked proteins is caveolin, which has two globular domains connected by a hairpin-shaped hydrophobic domain. Caveolin is found on the cytoplasmic side of the membrane and can dimerize to force an inward curvature of the membrane, forming small caves in the surface of the cell known as caveoli. Caveoli function in membrane trafficking and signal transduction. The formation of membrane curvature is important to the fusion of two membranes, which is mediated by integral proteins called fusion proteins. For example, in the synapse, action potential triggers the release of neurotransmitter by the presynaptic neuron. This is an exocytosis process that requires the neurotransmitter-filled secretory vesicles to fuse with the plasma membrane of the presynaptic neuron, mediated by fusion proteins called V-snares and T-snares. V-snares are found in the secretory vesicle, while T-snares are found in the plasma membrane. During fusion, V-snare and T-snare bind to each other, and the protein SNAP25 helps form four helixes that draw the membranes closer. The zipping of the helixes causes curvature and lateral tension on the bilayers, resulting in hemifusion between the outer layers of the two membranes. As the lateral tension increases, the two membranes completely fuse, creating a fusion pore that widens to release neurotransmitters within the secretory vesicle. Lastly, transmembrane cell adhesion proteins attach cells to each other and carry messages between the extracellular matrix and the cytoplasm. Integrins are cell surface signal receptors that carry signals in both directions across the plasma membrane. Cadherins are surface adhesion proteins that form homophilic interactions with identical cadherins in adjacent cell. Selectins bind specific polysaccharides on the surface of an adjacent cell and plays an important role in blood coagulation.